Module 6, Cell Cycle, Part 2.
So in the first part of this topic, we focused on some events that happen during mitosis, and in particular, looking at the function of the microtubules during mitosis. But for this part, we are going to concentrate on interphase, which covers the G1, the S, and the G2 phase of the cell cycle. So essentially the rest of the cell cycle. And we are going to look at this elaborate and biochemical control system that regulates the process of the cell through the cycle. So what is interesting about the cell cycle control system is that there are a series of checkpoints along the cell cycle that ask questions about the environment and condition of the DNA in the cell. So for example, there is a checkpoint as a G1 checkpoint just before entry to the S phase that asks, is the cell big enough? Is the environment favorable? Is the DNA damaged? There is another checkpoint in G2 that asks, is all the DNA replicated? Is it replicated properly? Is there any DNA damage? And there is a checkpoint in mitosis that asks, are all the chromosomes aligned on the spindle? How is the environment? On this slide, there are 3 checkpoints which are shown, but in reality, there are more than 3 checkpoints, and you will see in a moment. But the other interesting thing about the cell cycle control system is that it is a biochemical system. So it is regulated by proteins within the cell. And here is a classic experiment that supports the hypothesis that the cell cycle control system is actually a biochemical control system. If you take a cell that is in mitosis and fuse it with a cell that is either in the G1 or S or the G2 phase, there's a lot of interesting ways to do this. You can do this by electroporation. You can do this by incubating the cells with a polyethylene glycol that causes the membranes to fuse. Regardless of the method that you use, you end up with a heterokaryon. So this is a cell with double the amount of DNA. What you will notice in this heterokaryon, in this fused cell, is that the DNA that was originally in the nucleus of the G1 or the S or the G2 phase cell, the DNA begins to condense. So you see condensation, and that tells us there is some kind of a factor in the mitotic cell that can induce these condensation of the DNA from the G1, S, or G2 phase. And for a long time before the scientists discovered what this controlling protein or controlling factor was, they called it mitosis promoting factor, or MPF. As it turns out that MPF is a dimer that is made up of a protein known as cyclin and another protein that is kinase called cyclin-dependent kinase, or CDK. And CDK is a serine and threonine kinase, which means it phosphorylates the serine residues and the threonine residues on other proteins. So here is a very simplified diagram of how these CDK cyclins work. So what happens in these dimers come together at various points of the cell cycle, and when cyclin is bound to the CDK becomes activated. So you have the CDK cyclin pairs that operates in mitosis. You have the CDK cyclin pairs that operates in G1. Then you have the CDK cyclin pairs that operate in the S phase. And so you have a number of CDK cyclin dimers that operate throughout the cell cycle. And the reason why they are called cyclin is because the cyclin portion of this dimer goes through a cycle of synthesis and degradation with every single cell cycle. So with every single cell cycle, you get synthesis of these new cyclins, which combine with the CDKs in the cell to activate them at various points of the cell cycle. And here is just another way to look at it, at the activity of the CDK cyclin. The CDKs are active only in stages of the cell cycle they trigger or activate. So you have the G1 CDK cyclin, the S phase CDK cyclin. Here you can see that activity rises during the G1 phase and then drop off during the S phase. So you have additional S-phase CDK cyclin dimers that are active in the S phase, shown here in the red, and their activity drops off in the late G2 or during mitosis. You have the mitotic CDK cyclins that operate during the mitosis, and also shown in this slide is the activity of this APCC, which is an ubiquitin ligase. The APCC complex ubiquitinylate the anaphase inhibiting protein and its degradation through the proteasome. Please recap that proteasomes are large complexes of protein that will degrade other proteins that are ubiquitinated, which is going to become relevant in a minute. But you can see that the activity of the APCC, ubiquitin ligase, increasing during the mitosis and then decreasing, shown here in the green, at the G1 and the S phase. And this is a very basic overview and is the only mechanism that operates during the cell cycle, as you will see in a minute. Also, another important thing that is relevant, that although we mentioned here that these are G1S CDKs and the cyclins, S-phase CDK cyclins, they are also in the metazoans named as cyclin D, cyclin E, cyclin A's. And just for simplicity, we will mention them mainly by the phase they operate, like G1 S-phase cyclin or S-phase cyclin CDKs or mitotic CDKs. So here is what it looks like at the molecular level, and it seems complex, but it is not as complex as this slide would lead you to believe. And we'll just go through this step by step, so you can appreciate what is happening at each stage of the cell cycle. So we're going to start at number 1. So at number 1, the cell is either in early G1 or mid G1 phase, and DNA pre-replication complexes are already starting to assemble at the origin of replication. There are many origins of replication per chromosome, just not one. So you should remember from the biochemistry that when DNA is replicated, it begins at origin of replication, where large protein complexes, the replication complexes, assemble. This occurs at origin of replication to initiate DNA replication. And these are actually already starting to assemble in G1. So the cell is getting ready to replicate its DNA in the S phase. Now we are going to skip the number 2 and go to number 3, because number 2 is actually a leftover from the mitosis. And we're going to come back to that when we come around the cycle. Okay, another thing that is happening in the mid-late G1 is you get the activation of G1 cyclin CDK dimers. And when they become activated, they actually increase the expression of the S-phase cyclin. So the G1 phase cyclin induce the expression of S-phase cyclin, which are going to play a role in the DNA replication, as you can see in a minute. These S-phase cyclins are actually made during G1, but you don't want them to be active in G1, as it turns out that there is an inhibitor that is present during the G1, and this S-phase cyclin CDK dimer interacts with the inhibitor, and it remains inactive until the cell is ready to move into the S phase. The other job of the G1 cyclin CDK dimer is to eventually phosphorylate this inhibitor. Phosphorylation of the inhibitor is a signal for ubiquitination, so ubiquitin groups are added to this inhibitor by ubiquitin ligase SCF. Just for your information, SCF is named after the constituent proteins like SKP1, choline, and F-box protein, but that is not needed for your quiz or exam. Anyway, once SCF ubiquitinates the S-phase inhibitor, it becomes a target of the proteasome and it's degraded, as shown here in number 5. And the inhibitor is degraded, so the S-phase cyclin becomes activated and the cell enters the S phase. So this S-phase cyclin CDK dimer is very active during the DNA replication, and you will see in a minute that it phosphorylates a number of factors on the replication complex, which activates the complex. And the cell passes through the S phase, and the cell then enters the G2. In G2, the mitotic cyclin CDKs are synthesized and becomes activated, and then the cell can enter the mitosis. Now, we are not talking about any of the checkpoints just yet. In a minute, we are going to talk about how the cell can halt the cell cycle if there is a problem. But right now, we are just talking about a normal cell going through the cell cycle without any problem. So now the cell has entered the mitosis, and at metaphase, you remember from the previous part of this topic, one of the very interesting visual changes that you see in the cell is the alignment of all the chromosomes at the center of the cell. So all the chromosomes are attached to the kinetochore microtubules. All the cell move from the metaphase to anaphase. A second proteasome complex becomes activated, and this proteasome complex is responsible for degradation of a protein known as securin. And securin is one of the proteins in the cohesion complex that is holding the two sister chromatids together. And of course, the degradation of the proteins in that complex is necessary because during anaphase, the results of shortening of the kinetochore microtubules, and as a result of forces that are imparted by the polar microtubule and astral microtubules, you have the separation of the sister chromatids and their movement towards the pole of the cell. Right after anaphase, you have the activation of another proteasome complex, and that proteasome complex is responsible for the degradation of the mitotic cyclin CDKs that are hanging around. And at this point, the cell can enter the telophase and finally the cytokinesis. So let's make a full circles here and go back to step 2 here. Another job of the G1 cyclin CDK complex is to inactivate some of the component of the proteasome here that is acted in the step 9, because you don't want this proteasome to be active at different parts of the cell cycle, because it might degrade the other cyclin that are not used during the mitosis that could be detrimental to the cell cycle. So what I want to look at more closely is labeled here as 1, 2, and 3. So first, I want to show you how the G1 cyclin CDK dimer can actually activate the expression of other genes. To see how it does this, we will look at this at the molecular level. Then we are going to look at the molecular level how the inhibitor becomes phosphorylated by the G1 cyclin CDK dimer. And then the last thing that I want to look at in detail is how the S-phase cyclin CDKs actually initiate and regulate the DNA replication during the S phase. Okay, so here are some examples of the G1 cyclin CDK dimers, represented on this diagram as the cyclin D CDK4, and another G1 cyclin CDK dimer is cyclin D CDK6. So I want to point out on the previous diagram, it looked like there was only one G1 cyclin CDK dimer. There's actually a bunch of different ones here, but to simplify the diagram, there is only one listed, but there are many of them. So there are cyclin D CDK4, there is cyclin D CDK6, and there is a third cyclin, cyclin E CDK2. You don't need to know the specific names of all the cyclin and the CDKs, but you should be aware that there are multiple G1 cyclin CDK complexes that are operating during the G1 and during the transition from the G1 to S phase. Also shown here is another dimer made up of two proteins. One protein is known as RB, and RB stands for retinoblastoma. And the other protein is E2F, which is a transcription factor. And when RB is bound to E2F in this fashion, E2F is inactive. So this is an inactive protein complex. As cyclin D is being synthesized in G1, and as the cyclin D CDK dimer becomes activated during the G1, one of the targets of this kinase is the retinoblastoma protein. So the RB becomes phosphorylated. And so this creates a conformational change in the RB such that it releases the E2F transcription factor, and E2F transcription factor can bind to upstream region of genes and initiate gene expression of a wide variety of genes. One of the genes that it increases the expression of is itself. So it binds to the upstream region of its own gene, turns itself on. So you can get even more E2F. Another gene that E2F induces expression of the cyclin E. So you get a synthesis of a second G1 cyclin CDK dimer that becomes activated, and it can actually feed back into the process and phosphorylates more of the retinoblastoma to further activate E2F. So this is what it looks like in the molecular level. So here you have RB and E2F. The RB is inhibiting E2F, but when the RB becomes phosphorylated by, for example, cyclin D CDK4 or cyclin D CDK6, you get the phosphorylation of RB, the release of E2F, and E2F becomes activated. And then it can drive the expression of other cyclins and other genes. And some of these genes include the genes that are necessary for DNA replication during the S phase. And there are some proteins that actually mediate the centrosome duplication, which is going to become important later when the cell divides. So then, of course, the cyclin E CDK2 or cyclin A CDK2 can feed back into the process and phosphorylate even more RB to make the E2F work much more, to increase the transcription level. And the retinoblastoma protein is interesting. It's so named because it was discovered in the context of cancer. So retinoblastoma is a cancer of the eye, specifically the retina, and patients with retinoblastoma have a mutated RB that is non-functional. You can see in this image that the white yellow mass or tumor, which is visible through the pupil. Usually retinoblastoma is diagnosed before a child reaches the age of 3. The retinoblastoma makes complete sense in the context of cancer because remember, cancer, we are going to talk about this more when we talk about cell biology of disease and cell biology of cancer. Cancer is just unregulated cell growth. So you have this very complex cell cycle control system, and when something goes wrong, you can get uncontrolled replication of cells, which forms a tumor. So this is what is happening in the case of retinoblastoma. Individuals who have retinoblastoma do not have a functional RB, and since they do not have a functional RB, the E2F is always active. And that's why it is driving the cell cycle to progress into G1 to S phase. And when we're going to talk more about the retinoblastoma, when we talk about cell biology of the cancer in the last topic of this course. All right. What about the S-phase cyclin inhibitor? And remember that I told you that E2F can induce the expression of S-phase cyclins, but you don't want the S-phase cyclin CDK complexes to be active during G1. You only want it to be active upon entry into the S phase. The S phase cyclin CDK complexes begin to accumulate in the G1 but are inhibited by a protein known as SYK1. So SYK1 is an inhibitor of the cyclin S-phase cyclin CDK dimer. So eventually one or more of the G1 cyclins will phosphorylate the SYK1, and when SYK1 becomes phosphorylated, it becomes a target for ubiquitination. There is the addition of ubiquitin groups which tags the SYK1 for degradation by a proteasome, and so it becomes degraded. Then the S-phase cyclin CDK dimer is activated and promotes the DNA replication in the S phase. And this is exactly how the S-phase cyclin will promote the new replication. This is a very complicated slide with a lot of proteins on it. I'm just going to talk about it in general. So you should remember from biochemistry that I mentioned this already in this part of the subject, that you have these pre-replication complexes that assemble at the origin of replication. And it usually happens in the early to mid G1. And some of these are going to participate in DNA replication. Some of these components are inhibitors of DNA replication. Some of these are inhibitors that are preventing the action of these proteins that are involved in DNA replication. Once you get an active S-phase cyclin, which is shown here in the step 2, it will phosphorylate a number of components or proteins of the origin replication complex here in the blue. You can see that these all yellow phosphate groups have been added. In some cases, this will cause a conformational change in the protein such that they fall off the replication origin and you no longer have inhibition of that replication origin. So now the DNA begins to unwind and phosphorylation groups are added directly onto the components of the replication origin, which activates them and will induce the initiation of DNA replication in the cell. Again, this is just generally how the S-phase cyclin initiate DNA replication. Obviously, it is a much more complex than what I just discussed, but just the overview of the process is necessary. So, as I mentioned at the beginning of the segment that there are a number of checkpoints and the cell can actually halt the cell cycle at these checkpoints if necessary. So there are 4 DNA damage checkpoints. There is an unreplicated DNA checkpoint shown as number 1. The unreplicated checkpoint happens at the end of G2 right before the cell enters into the M phase. There is a spindle assembly checkpoint that happens in mitosis, that's number 2. There is a chromosome segregation checkpoint that also happens during mitosis, number 3. And then 4 DNA damage checkpoints that are scattered throughout the cell cycle. So let's look at each of these individually. So when the cell comes out of the S phase and enters the G2, you would hope that all the DNA is replicated properly. And so there is an unreplicated DNA checkpoint. And in other words, the cell is asking itself, is all my DNA replicated? And if it is not, a protein known as ATR in turn can activate a protein known as CHEK1. CHEK1 can inhibit another protein known as CDC25C. And CDC25C is responsible for activating the cyclins that mediates the G2 to M phase entry. So if there is an unreplicated DNA, the cell can halt the cell cycle before the entry to mitosis through this system right here. Right before the cell enters into the M phase, there is a spindle assembly checkpoint, number 2. So the cell is checking whether the spindle is formed properly, whether all the chromosomes are lined at the center, and if they are not, a protein known as MAD2 will inhibit the proteasome that is responsible for degrading the securin. Remember, securin is one of those proteins that keeps the 2 sister chromatids together. And so if securin cannot be degraded because this proteasome is inhibited, the cell is not going to enter into the anaphase because the cell will not be able to pull those sister chromatids apart. Another checkpoint is known as the chromosome segregation checkpoint, which happens right before telophase. And if the chromosomes are not delivered to the poles properly, if they are not segregated properly, they are going to get inhibition of protein known as CDC14. And CDC14 is responsible for activating these proteasome complex, which degrades and ubiquitinates some of the mitotic cyclins. So if this checkpoint is activated, the cell would not enter telophase at this point. There are 4 DNA damage checkpoints that are very interesting because they are all regulated by the same protein, which is known as P53. Interestingly, P53 is also known popularly as the guardian of the genome among cell biologists. So here are 4 checkpoints, and 4A is a checkpoint. This checkpoint is checking if the DNA damage occurred during the G1. 4B is checking the DNA damage just before it enters into the S phase. Then you have the 4C, checking the DNA damage during the S phase. And then you have the 4D, which is checking the DNA damage during the G2 phase. So what happens is that you get a protein called ATMR, which will activate P53. P53 will induce the expression of the P21 CYP. The P21 CYP will inhibit any cyclin CDK dimers present and halt the cell cycle until the DNA damage can be corrected. So say here is that this checkpoint, the ATMR, activates the P53. The P53 induces the expression of P21 CYP. P21 CYP inhibit the cyclin. Likewise, just before the S phase, once again, your ATMR activate P53, which induces the expression of P21 CYP, which inhibits the cyclin. And you also have the same process here, ATMR, P53, P21 CYP, and you have inhibiting this cyclin. And then you have right here before the entry into the M phase. Now, what is interesting is that the two of these DNA damage checkpoints have two processes that are monitoring whether there is a DNA damage. So right before entering into the S phase, these ATMR can also activate check 1-2, which will inhibit the CDC25A, which is normally responsible for activating cyclin. Likewise, you have over here, the ATMR can activate the check 1-2, which inhibits CDC25A, which is again would activate this cyclin. And so two of these DNA damage checkpoints have an added level of security to them. And that makes sense, for example, because this is just when the cell's entering the S phase and when the DNA is actually being replicated. You don't want to replicate your DNA if you have damaged DNA. All right, so how does this P53 work? As it turns out that the P53 is a transcription factor and it is not very stable. So the cell makes it and it becomes degraded very quickly unless there is DNA damage. And when there is DNA damage, the P53 becomes stabilized and it can bind upstream to a region of a number of genes. One of these genes happens to be P21 CYP. So P53 induces the synthesis of P21 CYP protein, and it will also bind to any cyclin CDK dimer that is present during the cell cycle and inhibit the cell cycle. So it is a very powerful cell cycle control system.
So this concludes Module 6, Cell Cycle, Part 2.